Well, good evening and welcome everyone. My name is Agam Patel and I'm the Associate Director here at UCR's Palm Desert Center, and it is my privilege to welcome all of you to this third part of our four-part lecture series called Exploring Our Aging Brain. The lineup in this series is spectacular, and if you've missed the first two lectures in this series, fear not, we got you covered. Subscribe to our UCR Palm Desert's YouTube channel and catch up on all these informative lectures you may have missed. Before we begin, I'd like to thank the Palm Desert partners who are instrumental in contributing towards programming like this. If you'd like to learn more about our partners program, we'll put a link up in the chat shortly. I also want to thank the entire UCR Palm Desert team, without which none of this would be possible. So thank you one and all. All right, let's get started, shall we? This program is brought to you in collaboration with UCR's Aging Initiative. Moderating tonight's discussion will be Dr. Lonnie Bennett, an assistant professor of psychology at the University of California, Riverside, and co-director of the UCR Aging Initiative. She received her bachelor's degree in cognitive science from the University of California, Irvine, a PhD in lifespan cognitive neuroscience from Georgetown University, and completed postdoctoral research at the University of Texas at Dallas and University of California, Irvine. Her research seeks to advance our understanding of the way we acquire, retain, and retrieve information. She is particularly interested in how these learning and memory processes are affected in aging, and studies their neural substrates using magnetic resonance. Her research has attracted funding from the National Institute on Aging at every stage of her career. Welcome, Lonnie.
Thank you so much, Agam, for the wonderful introduction to myself as well as to this series. So as Agam mentioned, this is our third installment of our Exploring Our Aging Brain series. If you enjoy it, please stay tuned for our fourth and final session next month, as well as check out the first two sessions that are available online. Also, as Agam mentioned, the event tonight is sponsored, at least in part, by the UCR Aging Initiative. So I just want to tell you a little bit about our initiative, which seeks to create a world-class institute on healthy aging at the UC Riverside campus. And we envision this being a center of research, education, as well as a hub for our community. So our vision is to create a uniquely integrative program that considers aging across multiple levels, from molecules to individuals to society. And tonight's talk is going to be a great example of that. We're really interested in understanding the integral role of human diversity in aging. You'll see tonight that we are joined by our founding director, Dr. Aaron Seitz, as well as my co-director, Dr. Marcus Call. But the initiative actually involves more than 60 faculty and staff from over 7 colleges on the UC Riverside campus. For tonight, we are going to focus on pathways and destinations. We're going to be exploring healthy brain aging or age-associated dementia, the roles of genetics, life experiences, and chronic inflammation. I'm going to first introduce both of our wonderful speakers. They will then present their work to you, and at the end, I will moderate questions. So if you have any questions throughout the talk, please feel free to add them to the Q&A feed, or drop them in the chat. We'll be monitoring both. Okay, for our first of our 2 wonderful speakers, we are joined by Dr. Monica Carson. She is the S. Sue Johnson Presidential Endowed Chair in Glial Neuronal Interactions, a professor and chair of biomedical sciences at the School of Medicine at UC Riverside, director of the Center for Glial Neuronal Interactions, and editor-in-chief for the Journal of Neuroinflammation. She completed her PhD at the University of Pennsylvania and has been an active member of both American and international neurochemistry societies since her graduate school days. Her research focuses on identifying how interactions between the brain and the immune system support lifelong brain health, and how genetics, inflammation, and environmental insults trigger or exacerbate neurodegenerative disorders. Our second speaker tonight is Dr. Chandra Reynolds. She's a professor of psychology at UC Riverside. She's also a fellow of the Gerontological Society of America, the Association for Psychological Sciences, and the Academy of Behavioral Medicine Research. She received her PhD in psychology from the University of Southern California and completed postdoctoral training in gerontology, also at USC, as well as in behavioral genetics at the University of Colorado in Boulder. Dr. Reynolds' expertise is in genetic and environmental factors that contribute to cognitive aging from a lifespan perspective, and she leads multiple studies of individuals, twins, and siblings to evaluate life experiences and genetic influences that act together to shape health, well-being, and cognition. Please join me in welcoming them.
Thank you for that very kind introduction. And I'm going to now begin sharing my screen. And we go there. And I'm really delighted in this opportunity to participate jointly in this session with Professor Chandra Reynolds. It is an example of how our campus, I think, is very strong in communicating across disciplines and really coming together to see the whole picture, not just our slice that we're specialized in. And so what I'm going to lead off and talk about today is start the discussions on healthy brain aging or age-associated dementia. It's what's the pathways to get to these different destinations. All of us want to have the destination of healthy brain aging, and we want to not accidentally get on the pathway to age-associated dementia. And both Professor Reynolds and myself will be talking about three different aspects: genetics, life experiences, and the role of chronic inflammation in helping to determine where do we end up. And also, I think both of us are going to give you some messages that you, all of us as individuals, have the ability to control which pathway we're on and which destination we are going to, or how fast we will stay with healthy brain aging and how fast we will transition to age-associated dementia. So with that, I am also introducing to you that my part of this story is I'm really going to be talking to you from the perspective of this cell that lives in the brain. It's called the microglia. And when we look at these pictures in the background, these green things, those are actually these immune cells. It's the brain's macrophage that lives full time in the brain, and it is everywhere in the brain. There's not any part of the brain or the spinal cord that lacks microglia. So that means your brain and your spinal cord, your nervous system, has an immune system that works full time, always in the brain and the spinal cord. And we now know, to tell you the end of the story, that these cells are essential to maintaining brain health. But if things go awry, they will contribute to our transferring to the pathway of age-associated dementia. So when we think about aging, let's start at the beginning. What happens as we age? We start off hopefully in good health, but we always look at how our progression is going to go is where we start. What was our starting health? And, most people think of it that we have a peak and then we decline. And that is not necessarily the best way to be thinking about this. It is really appropriate to be thinking about our progression across life as that, a progression. We change, but we can have healthy aging. But some things are going to accelerate. Some of our life choices and our exposures are going to accelerate our aging, so we prematurely age, or we can actually go against the odds and slow the aging trajectory. This will be modified, how close you are to aging and disease or how close you are toward healthy aging can be modified by genetics. But really, it's amazing how much of this whole story is actually under our control. So what are we really afraid of when we're aging? most of us, myself included, I want to keep good brain health. And that means so much. I mean, that's like, I want to know that I am I. I want to know who my community is. I want to know what's going on. And so we want to avoid this thing called dementia. When we think about dementia, the first thing is understanding what is dementia. A lot of times we'll just go, well, it's Alzheimer's. I'm afraid of Alzheimer's. And without a doubt, as you get older, the risk for dementia increases, and the lion's share of the dementia that one is likely to develop is Alzheimer's. So by the time folks are in their 80s, of the individuals who have dementia, really over half of them have Alzheimer's forms. But there are all these other forms of dementia, and they have overlap, but they are not identical. So that's something to become aware of. And some of your life experiences, some of your genetics, and some of your choices are going to predispose you to one or the other. And some of these, tend to have more genetic relatedness, and some of these are associated with very specific infections that we might have. So let's think a little bit more about dementia. And one of the first things to think about is, what is dementia not? And what is healthy aging? First and foremost, we have to embrace the idea it's possible to age in a healthy fashion for a majority of our life. For most of us, some of us are going to have a tougher time with it because of some of the genetics that we had, or some of the choices that happened early in our life. But when we look at healthy aging as the goal, we have to see that dementia is the aberration. It is a disease state. So when we think about, the senile old fool, which I hope I am not, when we look at senile, that in colloquial terms is what most folks clinically are looking at as dementia. But let's again look at what dementia is not. Normal aging does not necessarily result into senility. That's the first thing. So normal aging doesn't mean you have to become demented. Do we change? Does our brain function change as we age? Yes, it does. I can say that when I compare myself now, and I am now well above the age of 50, I can tell you that at this point, and I compare myself to younger, I'm more forgetful than I was. So you compare self to self, you have an increase in forgetfulness after the age of 50 that's really noticeable. But there's a lot to it. Yes, you have a lot more to remember, but there's also some other things that are going on. You have slowing of response times. That is going to be happening. But let's also think that there's speed accuracy trade-offs. If you actually compare self to self, and I can say this, I often would talk to my children as they were growing up, and I say, in what world did this make sense? Well, I could almost say that to my younger self. In what world did something make sense? When I'm confronted with a set of facts. Earlier stage in my life, I was faster at doing things. I could remember them faster, but I didn't have as good pattern recognition. And so there is the saying that wisdom comes with age, and there's some truth in it. You have more to remember, you have more life experiences, and you're seeing whole patterns that really you could not see earlier when comparing self to self. So I think it's all a very important thing that when we think of aging, we do know our brain is functioning. We need to have both types of function in the population for our species to survive. We need what we were good at as young. We also need what happens with our brain and the skills that we acquire as we age. So this is part of normal aging. But let's think about then, what is dementia? I said there's multiple different types of dementia, but when we're looking at that, when we're looking at things, you can't just say it's poor brain function. Dementia is loss of function. So whatever function you had, it could be high, could be middle, could be low for any individual. Dementia has to be a loss from what you had. And it's going to be in various domains: your ability to learn, memory, your ability to use language and things like that. It's called executive function, your ability to coordinate a lot of things. Complex attention. I'm demonstrating that now. I'm actually thinking ahead what I'm going to say. I'm paying attention to my actual slides. I'm looking at the chat to see if anything's come up. So, complex attention. I can pay attention to more than one thing at a time and integrate them. Dementia, another thing people don't often realize with dementia, sometimes you're going to have problems with perception. In your eyes and your ears may actually be working, but you don't know what to do with that information that you're taking in with your eyes. You may see something, but you can't quite interpret it correctly. You may hear things, you may hear words, but you can't quite understand them. Similarly with social cognition, dementia could be, your ability to interact with others may change and become less good and less effective than it was before. So these are differences. I bring this thing that was actually found on the web a couple of years ago that I thought was a really nice comparison between normal memory loss versus dementia. And you can see that in normally, as we get older, we forget someone's name, we forget a word during a conversation, but we remember later. It's like it's on the tip of your tongue, you can't do it. But dementia is a much more extreme, losing oneself in the conversation. And I mean, not just minorly losing yourself, but stopping abruptly, forgetting what to say next, repeating oneself often. Normal is forgetting an appointment from time to time. Who doesn't? Dementia is forgetting recently learned information, having to ask for it over and over again repetitively, increasing reliance on others to help remember to complete tasks. Similarly, it's normal to forget what day it is occasionally. It's a long weekend. You might be retired. Like, what day is it actually? But dementia is completely losing track of the time of the day or the season, forgetting where you are and how you got there. It's normal to get irritated when one's normal routine is altered. By this time of my age, I know what I like. Why are my adult kids coming to home and disrupting things? That's normal. Dementia is getting extremely agitated by a change in routine, especially when the agitation is not normal for your personality type. So it's a real change in what you could really like or dislike. We all make bad decisions, especially when it comes to money. That's normal. Dementia is consistently showing poor financial decisions making ability. It's normal to feel tired, want to rest, stay home occasionally, but it's not normal to remove oneself from your favorite hobbies, to completely, because you forget things, it's difficult to complete. It's not normal to become isolated and withdraw and not to venture out. You can lose items occasionally, normal. Putting items in strange places, and please don't be freaked out when you're putting away your milk and you're putting away things and you accidentally put maybe something in there was your hand in the refrigerator. But it's when you purposefully you're putting these things away and you're thinking that, my keys do go into the refrigerator. That's a different kind of thing. And vision changes due to cataracts. Yes, my eyes are not as good as they were, but it's, again, that difficulty understanding what I'm seeing. So these are dementia. So let's go back to the fact of, and this is all these deficits we have to look at. It's not because you had a vitamin deficiency, not because of something else that had to do with depression. These are really organic changes that are causing. That's dementia. So when we look at this, a lot of us are going, I can see that I'm living with somebody who has that, or I've seen this in my family. It's my mom, my grandma, my granddad. Everybody seems, to get a little senile, to have these things. And so a lot of folks are thinking, I just have bad genes. I have bad this. Do I just give up? What do I do? Is there any hope to deal with stuff? Well, we might have risk factors. That is without a doubt. You might have risk factors for cardiovascular problems. You might have risk factors for various types of cancers. But especially when we think about risk factors, once we identify we have risk factors, if we have genetic types of things, we're going to do things that minimize them. If you have cardiovascular problems, well, you're going to start to do things like, maybe I should watch my weight. Maybe I should make sure to be doing exercise. And so in that same way, we may have genetic risk factors, but there are things that we can do about them. And the other thing to point out is when we follow things in a family, often it's not actually the genes. Often it's you either live in a common environment, you're next to the toxic dump, you have, you're next to very poor air quality. That's common. Or more commonly, y'all like the same foods. I know in my family, I come from a very diverse family, but we seem to really love our holiday foods. And a lot of, I think, our weight issues in our family are not genetic. It is our embracing our common heritage in cooking. So these are the things one has to start looking at when one looks at things in a family. Have we all done the same things? Maybe if I change some of my family behaviors, will I minimize any genetic risk I have? And maybe there was no genetic risk. Maybe it was totally lifestyle. Now, when we start talking about brain immune interactions, and we're talking about things that can influence which destination we go on and which pathway we're on, pathway to healthy aging, pathway to dementia, we start looking at the brain and the immune system are hardwired for communication. And I start off showing you this picture of a brain, and it's green. It's labeling these neurons, these things, these little blobs there are cell bodies, and then they have their axons that are reaching out. Now we stain that same thing, the same brain, to label the blood vessels. You see big blood vessels, you see little blood vessels, they're everywhere. And when you overlay them, you look, the neurons and the blood, they're totally next to each other. And that's something to think about. If you have, ideal body weight, your brain is only 2% of your total body weight, yet it uses 20% of your blood. So this tells you how important vascular health and heart health is to keeping your brain healthy. And it also tells you that the immune system, which lives a lot in the blood, has constant access to the brain. It's going to be going through. And that's in addition to those microglia that were the full-time immune cells that lived in the brain. And one of the things that I always point out is when you have this vague sense that things aren't right, we now know actually amongst the many things that are contributing to your success is your immune system reporting back to your brain that it sees the parts of the body that aren't well. It doesn't say, yeah, it's your foot, it's this. But we have molecules, these things called cytokines, that communicate to the brain, and the brain starts realizing, often through the microglia that communicate to the neurons, some of these other glia called astrates can communicate to the neurons. We have the body via the immune system telling the brain things aren't good, and it changes the brain. And in fact, one of the things we can see that changes the brain is something that happens when you get sick. When you have inflammation outside your body, you actually change a whole bunch of behaviors. You start like, I don't want to eat. I'm sort of tired. I don't want to interact with people. I don't really want to do anything. And most people said, of course I don't want to do things. I don't feel good. I feel punky. I don't feel good. Of course I want to do that. Turns out that's actually not true. It's actually your brain is telling you not to do those things. Yes, you don't feel good, but we can mimic that same behavior by injecting into animals and into people, because it's one of the therapies for multiple sclerosis, things that actually activate this system and make your body think there's inflammation. We can actually cause this behavior. So we know it's actually something that we determine. And so the question a lot of mental illness and neurodegenerative disease is when is this working for you? And you're going, why would that ever work for me? Well, when you're sick, you want to sleep so your body can take care of yourself. You don't want to eat because you don't want food available in your bloodstream for the bacteria or the viruses, and you don't want to get people around you sick. So that's a good thing. But when is this bad? Is when this becomes a little bit more permanent, when you have chronic inflammation. What's tipping the balance there? And we actually can't do without this interaction, because otherwise you'll have parasites, you have infections that would attack your brain, and brain death is our death. It turns out the immune system is important for repairing the brain, such as after concussions. And what's been learned in the last 10 years is the immune system actually helps with memory formation and actually helps with us learning new things. So like Goldilocks, we want just as much immune reactions, but not too little, and we want it just the right time. And this is where we start looking at the things that we choose to do. Environment, cardiovascular health, various kinds of things that happen to us, infections that we have, and consequence of substance abuse. All of these things, which we know are risk factors for dementia, they have in common that they cause chronic inflammation in the body. And this chronic inflammation we now know contributes to the process leading to dementia in the brain. And in fact, it's also contributing to some things which we now know are this brain fog that happens in chemotherapy and is also part of long COVID. So these are the microglia that I told you about. They're pretty. And here's a close-up on one of these cells. And here's another one wrapping itself around a blood vessel to help with the communication. And when we think about the microglia, they do a lot of things. They're just like moms. And I'm saying I respect dads. I expect aunts and grandmothers to take care of people, but I'm a mom, so I tell us from the mom's viewpoint. But all these folks could do these things. But moms, they nurture and care. They house clean and keep, and they protect the brain. And so that's what microglia are doing. And so when we think about this, and that's the pretty microglia again, we also think that when mom's happy, the family's happy. And why is that? Because mom's happy, the microglia is happy if everybody else is happy, because the microglia with all those processes is reaching out to each aspect, every little neuron. And just like a mom at a birthday party in a park, where if you've ever had to do that, and I've had to do that, you have like 10 kids and your whole day is going 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 1, 2, 3, 4, 5, 6, 7, 8, 10, 1, 2, 3, 4, 5, 6, 7, 8. Where did 2 of them go? That's what the microglia is doing full time, and it's performing these activities. So mom's happy when everybody else is happy and the family's happy. But sometimes, mom gets a little irritated. I got irritated and the rest of the family got irritated. And so mom's happy, everybody's happy. But when mom's not happy, pretty much everybody is on eggshells. And mom could be unhappy because there's a problem with mom, or mom could be happy because you're going after her kids and she's got a problem with you and she's accidentally got her kid a little bit more upset, but it's in the argument of defense. So we have to think about what's happening to do these different states. And one of the things we know that microglia, the moms of the brain, they notice what's happening to the kids, the neurons, and they remember. Here's a picture of healthy microglia, all nice and little. They have these processes. But after a concussion, you hit your head, traumatic brain injury, they get all big and angry. And they actually remember and don't go back to baseline for a long time. And microglia is their ability to put synapses together. And I always call it making play dates. Putting these neurons together is affected by how they have been asked to perform. So if they got activated, like we saw here with a concussion, they change how much they put things together or take them apart. And so that's what we have to think. Microglia are long-lived. Each of these microglia lives for decades. Pretty much the microglia that's in your brain, those showed up in your brain when you were in your mom's belly. So microglia remember trauma insults, which changes their subsequent reactions and responses. And here's a picture of what happens with traumatic brain injury. Here's a study where it's healthy individuals. You see it's all gray and purple. Well, in this imaging, inflammation is shown with things turning blue, green, yellow, and red. And you can see even 11 years after injury, there can be a lot of residual inflammation. The microglia are still activated. We can see that with inflammation, inflammation gets caused by some of our other common things, such as obesity. And so when we look at a picture here, green are microglia, and these are microglia in this part of the brain that controls a lot of our behaviors. And they look nice and small. They have these little thin processes. But all of a sudden, I look at this. They're all mad, and there's so many, and they bulked up. And this is the brains of males. And this is a rodent that we've done some experience with Dr. Costa's lab and Nancy Linez. And what's happened here is that when on high-fat diet that caused really huge obesity, you saw this inflammatory response in the brain of male mice, but you did not see it in female mice. What did that mean? Well, the obesity that we saw in the male mice was also associated with decreases in certain synapses in certain neuronal circuits. It changed the function of it. It actually changes parts of the brain that are controlling your feeding behavior and how you're reacting. So that's a big deal thing. So when we can also look at other things, our lifestyles and life exposures can also affect the brain, and specifically air we breathe. And I'm going to give you an example of things done by Joe Valdez and Paula da Silva Frost, who have been in my lab, about studies that they did in collaboration with the Low Lab on how exposures to airborne allergens, so these things that we breathe, actually change how the microglia are. And we've chosen to use a non-infectious extract of allergens from this fungus called Alternaria. Why? It's actually a real life major cause of allergies and asthma. It's the leading cause, actually. It's in the United States of severe asthma. And it's found in all of our houses. It's outdoors. It's everywhere. And again, you talk of green here. Here's untreated, a healthy, and here's after 1 week of exposure to these allergens sufficient to cause lung inflammation. And when we do this over time, we see this causes the microglia to change over time. And this is at 1 week, and this is at a month. And we can see that the females respond more initially, but they end up at the same spot. And in fact, when we look at things that are about the synapses, we again see that we have a change in the synapses. And we can see over long term, there's actually a long-term change in how the neuronal circuits in the brain are actually functioning and materially how they are. And what's curious about this and striking about this is specifically in the area that controls lung function. So definitely our exposures change things. We also can see that when we look at shape of these cells and we look at male mice, we see that we don't see any real change in the shape of the microglia in the male mice, but we do see a change in the female mice. And what we're seeing is that in the female mice, the microglia develop more branches. So it's like they can count, like the thing where I said counting how many kids are in the park. The microglia in the female mice were able to count more kids because they had more processes. And this is not how you respond in the brain changes over age. And so we said, look at, this is the same picture I showed you just a little bit ago. I just put them on top of each other. You had the healthy adult. You then gave that 1 week of airborne breathing exposure to the allergen and the microglia went crazy. But we did this in a young child in the mice. Nothing happened. So when we get exposures, actually affects things. So we actually didn't know before we did this who would be more responsive. So it's pretty striking that we get more responsive as we get older. And just to say something else, we've also done things where we've actually tricked the body to think that they had a bacterial infection. So we could just look at what was the immune response to a bacterial infection and what would be the effect in the brain. And we did this over the lifestyle. And this is from the equivalent of young children all the way to something that's not that old. It's like a 60-year-old individual in the mouse. And you don't have to pay attention to the actual graphs here, but the point that we see is that your actual response to bacterial inflammation in the brain of microglia and all these other inflammatory responses, they change quite a bit as you age. And the kind of allergens, somebody asked me an allergen question earlier, and I just saw that. We talked about that with a fungal allergen, which is actually in all of our environments. But here I'm talking about bacterial based inflammation. And again, we see even with bacterial inflammation, we're seeing different responses between males and females. And here, because bacterial responses may be more robust, we're seeing these globally throughout the brain. And just go for it. So microglia, they're the moms of the brain. when mom's happy, the family's happy. And when mom's not happy, well, yeah, the brain's not happy. And when we're thinking about therapies or when we're thinking about avoiding dementia and getting on the wrong pathway, we want to keep mom happy. Mom's happy because the family's happy. And when the family's happy, she's happy. If the family's unhappy, she's unhappy. And if mom's unhappy, the family's unhappy. So we have to start thinking about our choices, what exposures we're doing, some of the things about cardiovascular health, walking, all of these things are going to contribute to keeping us hopefully in the happy pile. So in general, I think what we want to do is we want to stay engaged. We want to remember diet, heart health, and we also want to sleep. And I just put the sleep because I haven't had a chance to go over that, is that sleep is important because sleep is like taking a sponge and just squeezing out all the junk that we sort of did through the day. And this is a thing called lymphatics. And we're actually squeezing it out, allowing all the junk to get to where immune cells live in our lymph nodes. They get educated about what they want to happen. So sleep is important. It becomes increasingly important as we age. Also staying engaged is important. So I hope that helps. And that when we think about this interaction of dementia, we do want to know that genetics does have an influence. They can predispose you to inflammation or poor cardiovascular health, but we have control about minimizing those aspects, minimizing inflammation, and these can and they do make a difference on aging well. And so with that, I'm going to turn it over and I will answer the many questions that are in the chat at the end of the second portion. So with this, I turn this over to Dr. Reynolds.
Thank you so much, Dr. Carson. I am very honored and happy to be able to partner with you on this third lecture of the series. Okay. I hope now that you can see my slides. So I am going to be talking about variability in aging from a different vantage point than Dr. Carson. And I'm going to be focusing on the variability in our aging, that we can see even individuals of the same age may show very different aging outcomes. One individual may be able to report the exact wording of a short story read over a year ago, and another individual may not recall what they had for breakfast that morning. And what leads to these individual differences in aging? Well, we can talk about what it means to successfully age, and that would include things such as high functioning across a number of our domains, including physical functioning, psychological functioning, and social functioning. And that includes, of course, intact memory performance and cognitive functioning. If you ask even young individuals what they fear most about aging, most will say that they fear the loss of cognitive functioning and memory over the changes they may see with physical functioning. In terms of our cognitive performance, we know that we, on average, across populations tend to have peak performance in our 20s and 30s for many abilities, including how quickly we can solve problems, for instance. Our peak for memory performance is about 20s and 30s, but our verbal abilities are the kinds of abilities that we keep showing increases on average with age. And it's not until 170s or so that we will see the beginnings of some declines for some individuals. But again, we find a lot of individual variability. And some of the work that I'm involved in is trying to understand what are some of the underlying factors that lead some individuals to maintain their cognitive functioning while others show more dramatic change. So one of the sets of factors that may be important to a certain degree are genetic factors. And we can infer the importance of genetic factors by comparing relatives who vary in terms of their genetic similarity. So we could compare, for instance, siblings to one another who share on average about 50% of their segregating genes in common, or if we're fortunate to have identical twins, we could compare their resemblance in comparison to fraternal twins or other siblings. So we can infer the importance of genes by comparing families who vary in genetic resemblance. We can also, of course, conduct DNA or genetic testing to get another lens on the more specific genetic pathways that might be important. Of course, our environments are so important for supporting or not how well we age. And we can look at this again in terms of family members who may be more or less similar based on the environments that they share. Certainly siblings who are reared in different homes, if there are adoptive siblings, for instance, we can get a lens on environmental factors that may contribute to similarity. But we also know that, for instance, identical twins, while they're identical genetically, may have very different lived lives and show quite a number of dissimilarities as well. And we can think about multiple levels of environments in the family home, environments in the neighborhood, as well, in terms of thinking about more proximal or close environments, if you will, and broader environmental contexts that may be important to how we age. So here is a pair of twins, and you can see some photos of them as they've lived in various periods of their lives. They participated in the octo-twin study, a study of identical and fraternal twins who were 80 years of age and older. I show them because of the nice feature of pathways that we're talking about tonight in terms of what might shape how well we age, especially cognitively. Octo-twin is just one study of twins that's important to understand aging, but there are also other studies such as the Swedish adoption twin study of aging, where in fact there were twin pairs reared apart and reared together. And some of the data I'm going to show you on the next slide comes from the SATSA study. And here what this graph represents are just a random selection of some of the SATSA twins who range in age from 50 to 90. You can see there are pairs of lines here that are color coded. And so we have, for instance, an aqua set of lines. One is for twin 1 and the other one is for their twin 2, their co-twin. And we have other pairs of lines here, again, color coded, that show their cognitive performance across time from when they started the SATSA study. So some of the twin pairs show very close patterns of performance, and other pairs of twins show quite dramatic differences in performance as they age, with some showing some accelerating declines. So these are a sample of identical twins, and in the panel below, we have a sample of fraternal twins who, again, some of the pairs show quite similar performance, but other pairs show quite different kinds of cognitive performance across time. And so we use this kind of data to get a handle on the extent to which genetic and environmental factors might shape our cognitive performance with age. In comparing fraternal and identical twins to one another on cognitive functioning measures, we can estimate on average about a contribution of genetic influences to why people differ in cognitive functioning of about 42%. In terms of the risk of late onset Alzheimer's disease, on average, there's about a 57% contribution of genetic influences on risk for Alzheimer's disease. Now, some of the estimates do vary across studies, so I'm just showing some overall picture across a number of studies. So again, we can say that cognitive functioning and risk of Alzheimer's disease are heritable, but you can also see that those estimates of how much genetic factors influence cognitive functioning and late Alzheimer's disease risk are not 100%. The rest of it is environmental influences that shape our cognitive functioning, performance as we age, and also risk of Alzheimer's disease. So a large role for the environment. In terms of the top measurable candidates or genes that contribute to Alzheimer's disease risk, we can see here on this word cloud 25 gene loci that contribute in a big way to Alzheimer's disease risk. We still do need to understand a lot of the other genes that are involved. This does not capture the full heritable influences that we see in the graph at the bottom right, but it does capture a lot of the Alzheimer's disease gene loci, and the size of the letters of the gene indicate the importance of their ranking in terms of contributions to Alzheimer's disease risk. And I think a takeaway point is across these 25 is most of these are involved in the cholesterol pathways and inflammation and immune pathways that Dr. Carson was discussing, in fact. And you'll see there's one that's highlighted in the biggest letters, which is APOE. The APOE gene codes for apolipoprotein E, which is the primary cholesterol transporter in the brain, and the brain is a big consumer of cholesterol. And when our brains undergo injury, in fact, there is this role for APOE to be a partner in some of the processes related to injury and repair. So again, APOE is a primary cholesterol transporter in the brain. It is implicated in cardiovascular disease and late onset Alzheimer's disease. There are 3 common versions or alleles that produce different versions of apolipoprotein E. And these are called E2, E3, E4, or epsilon 2, epsilon 3, epsilon 4. And the E4 allele is associated with cognitive change and increased risk of late onset Alzheimer's disease. It is about 17% prevalence in the population. Epsilon 3 is the most common of these 3 alleles. E4, or epsilon 4, the next most common, and epsilon 2, which may have a protective role, is the least common of these 3 versions. This epsilon 4 gene is associated with increased plaques in the brain vis-a-vis beta amyloid deposition, which is a component of senile plaques. So we've become interested in this particular gene in terms of its outsized role in Alzheimer's disease risk, though again, it's just one of many possible genes contributing to risk. In terms of looking at cognitive change, we've looked at, for instance, identical twins performance now here. So just focusing on our identical twins who share 100% of their genes in common, and looking at their memory performance, memory for facts. So remembering who wrote Huckleberry Finn, for instance, or what is the capital of Italy, for instance. We've looked at this performance over age, and again, age is showing on the bottom axis, and the performance level is shown on the vertical axis here. And again, each pair is color coded just to give you a sense. So we have this green pair here where one individual is performing better than their co-twin, although they are showing some declines. Again, the picture is that some of these twins are performing better while some of their co-twins are showing some losses with age. And again, given that they share 100% of their genes in common, what might be contributing factors? Well, of course, the environment comes into play, but it turns out to be interestingly complex. So for example, if we look at the differences in those aging profiles for semantic memory, and we look at whether or not the pairs carry one or more of the epsilon 4 alleles versus those identical pairs who don't, we can see, for instance, that the differences between the memory profiles, memory for facts here in this case, look different if you compare pairs who carry one or more copies of the epsilon 4 versus those pairs who don't. And it turns out that identical twins who carry the APOE4 allele show much more similar patterns. This is the extent of their differences. It's much smaller, the extent of their differences in the memory profiles, than identical twins who do not carry APOE4. We looked further into this, and one of the interesting findings is that identical twins without the APOE4 show different memory change profiles if one of the co-twins experiences more stress than their co-twin, then the differences between their memory profiles becomes much more different than their co-twin. So this suggests that we may have some differential sensitivity to environmental exposures that is in part related to genetic influences. Now, what about this APOE gene? So it does predict late onset Alzheimer's disease risk. Does it show any sort of interesting pattern with respect to cognitive functioning early in life? So how early might we see some potential influences? In another study of both twins and siblings, we have looked at cognitive performance in childhood at ages 7, 12, and 16, and looked at the extent to which having one or more copies of the APOE epsilon 4 E4 allele might be associated with differential performance earlier in life. And these are data from IQ batteries taken in the total sample, then by males and females. And what we can see is that carrying one or more copies of the APOE4 led to less optimal performance, although the performance is still quite high in all of the groups. So some small difference in performance. And it mainly was observable when we looked at males versus females among the female individuals. And again, this is early in life. So there may be some early vulnerabilities. It's a small effect, but it may be salient. And this is, of course, the age period where obviously the individuals are in school. And we hope that during this period, they are laying down essentially cognitive reserves that will help maintain functioning later in life. And so finding these differences early may have implications for earlier interventions, for instance. Now, I've talked about this differential sensitivity to the environment on the basis of genetic factors, and we have looked at genetic biomarkers of responsiveness to the environment as another way to look at such aspects. And so one thing that we know about the way in which our genes are expressed is that there may be some brakes placed on the expression of genes due to environmental exposures. And one of these kinds of factors is methylation. So this methylation process is a way in which a tag is placed on a sequence of DNA that alters the extent to which a gene might be expressed. Methylation as a process alters the expression of genes, it varies across different tissues in our bodies, including brain, blood, as well as muscle and other tissues. And it varies across the lifespan. And most importantly, it's not identical in identical twins. So our lived lives may have impact essentially on the extent to which we might find different genes being expressed or the extent to which they're being expressed. So for example, these are two plots. On the left is a plot of identical twins, and the plot on the right is a plot of fraternal twins. And for the identical twins, we're looking to see if we can predict their methylation pattern between twin 1 at age 69 and twin 2 at age 79, so 10 years later. And you can see that we get pretty good prediction of understanding gene expression for this particular methylation site for identical twins, and still somewhat good prediction if we look at the fraternal twins in terms of predicting their co-twins expression profile 10 years later, but much better for our identical twins. And this particular site happens to reside in the HLA region on chromosome 6 and is involved in immune function pathways. This other methylation site shows the pretty equal similarity for identical and fraternal twins. This site is in an aging pathway related to insulin-like growth factor. So we have looked at these patterns across over 350,000 methylation sites across the epigenome in twins at 2 time points. And some of the overarching features or findings that we have is indeed methylation as one biomarker of responsiveness to the environment shows some heritable influences, about 24% across those 350,000 plus sites at 69 years of age and 18% at 79 years. So some small heritable contributions, but they do vary. And for the sites that show the greatest heritability, we see that those sites showed enrichment in being involved in immune and inflammation pathways, as well as neurotransmitter activity pathways. So for about 5,000 sites that showed the strongest heritable component, we see that they're involved in inflammatory pathways and stress response pathways, for instance. So again, this suggests there may be some genetic contribution to responsiveness to the environment that's important to our aging outcomes. And then one of the last slides I will show before concluding is coming back to brain health and how much our health behaviors matter to how well we age. And so in looking at brain age, we can look at the predicted age of an individual's brain age versus aged match individuals. And there are certain features of the brain that go into the calculation of brain age. And these things include cortical thickness and subcortical volume data to create composite measures of brain age and compare to, again, a series of matched individuals to place the brain age of an individual. And we can look at the differences then between one's chronological age versus their brain age as an outcome. And in this study from the Vietnam era twin study of aging, a study of aging male twins, we have observed, for instance, a prediction of brain age from smoking at age 40 to brain age at ages 56 through age 68. And the findings suggest that smoking was associated with older brain age, smoking at age 40. And alcohol consumption at age 40 was also predictive of older brain age. Smoking, the smoking pathway was consistent with an environmental effect when we looked within identical twin pairs at which one smoked more than the other. The twin who smoked more had an older brain, and that was true both for identical and fraternal twins. So this was consistent with an environmental effect. In concluding from this series of findings, I've shown there may be some early life factors that contribute to cognitive aging that need some additional follow-up to understand what those early life vulnerabilities might be and how they can be buffered. There is an evidence for a dance between genes and environments for cognitive traits, that cognitive performance not only differs by genetic factors such as APOE, but we may be differentially sensitive to exposures that are stressful. And I've showed you some data from semantic memory change, but we have some other relevant findings as well that are consistent with that. And then as a biomarker of this sort of gene environment interplay, DNA methylation may point to the interaction between genes and environmental exposures such as stress. And we see a role for immune and inflammation pathways. And last, of course, health behaviors matter to both brain and cognitive aging. And to follow up a point from Dr. Carson, there's room for how we age, that we have some control over how we age in these pathways to either healthy cognitive aging versus dementia. So I'll end it with that and turn it back over to Dr. Bennett.
Thank you so much, both Dr. Reynolds and Dr. Carson. That was a pair of fantastic talks. I can see that we are a little bit over time. We'll stick around for a couple more minutes to answer some questions. I can also see that Monica Carson has been frantically answering your questions in the Q&A. I don't know, Dr. Carson, if there's any of the questions that you want to bring to a discussion for the group, or if anyone wants to drop any additional questions for Dr. Reynolds in the Q&A, I'm sure she'll be happy to answer them.
The only thing I'd like to say is I think what Dr. Reynolds has shown is tangibly the epigenetics is showing how these life experiences caused those changes. And she's been showing all the things I was showing on a level in model systems and on correlations, she's actually showing in human beings. So it's very profound what Dr. Reynolds is doing, and it's very difficult research to get. So I think we just want to have a real appreciation for the nature of the difficulty, because that's why it's been hard in newspapers and this, because people will just say, it seems to me this causes a problem. And the careful work that Dr. Reynolds is actually showing what are the risk factors that are accelerating aging.
Absolutely. I see that someone has raised their hand. I don't know actually if we're able to answer questions that way. We can give it a try. We've not done this before. So Patricia Macklin, if you're able to unmute yourself. The other option is to just drop your question either in the chat or the Q&A.
I think the individual, I know there's somebody staffing this who has a host function, might be able to unmute them.
Okay, I did it. I unmuted myself.
Great.
Okay, now, if someone is diagnosed with Alzheimer's, what is the best environment for them to find, in which to find themselves? Less stress, or just sit and watch TV all day, or play cards all day? What would you suggest for the environment of someone that has recently been diagnosed?
I don't know if Dr. Reynolds wants to answer that, but I can start it. Dr. Reynolds, did you want to take that, or?
Feel free, Dr. Carson, I'll follow up.
I think one of the things we're guided by is what are the risk factors for Alzheimer's disease? Because honestly, Alzheimer's disease starts about 15 years before you actually see the overt signs, because we've been having this chronic inflammation. So a lot of what we think of as risk factors are things that are actually accelerating the progression and reducing brain health. We know that engagement fosters brain health And lack of engagement fosters higher risk and faster progression for classic dementia. And that's why it's so important to check your hearing. If you start off as a hearing individual, if you're deaf, you have other things that you communicate with. But those of us who started off as a hearing community, then loss of hearing, we disengage and we don't have engagement, and that actually fosters progression. So keeping hearing good is, and keeping interacting. So I would say engagement and interaction, and yeah, the classic use it or lose it. Dr. Reynolds?
So I would just, of course, wholeheartedly agree with that. Engagement matters. And I think for the individual, doing things they have always enjoyed is really important. And I think trying to create a situation where individuals can still do those things that they like to do is very important. And social stimulation is still important for trying to maintain as long as possible functioning. So the example of watching TV all day, I think would be less optimal, than trying to create situations where people can interact still with their friends and other loved ones, for instance, and trying to maintain that as much as possible, the things that they have loved so much across their lives.
And what if they happen to have a spouse that yells at them constantly?
Well, I can't, of course, speak to this specific example, but we certainly know that stress is never optimal for aging, and so I think that that's a situation in which one would want to try to increase positivity and lower stress for all of us is helpful for all of our aging.
There's a couple of last questions that I want to get to in the chat. We have just about 2 minutes. So if Jane Bark has asked if there's such thing as an anti-inflammatory diet.
I would say that is such a complex thing. What's marketed as anti-inflammatory because it's antioxidant in the test tube versus in your metabolism. We all have various different genetics of how we metabolize food. And so I tend to recommend people to go to a healthy diet for their own physiology that maintains their own body weight, that maintains the best blood sugar and endocrine balance. And so it's basically, what is your metabolism? So there's definitely, all candy diet is bad. I can say that, but I think it's hard to buy into a specific thing. It really is an individual kind of thing. And this also needs to be done with your clinician or with a nutrition advisor.
Mary Joy Barnett has asked what behaviors may contribute to healthy aging.
Dr. Reynolds, why don't you start with that one?
Sure. Physical activity, again, always in consultation with your physician, but physical activity is one of the most important. Social engagement is also very important in terms of maintaining functioning. So those are two things I would say, and certainly cognitive engaging, but do the things you like to do. Like, don't start doing Sudoku if you can't stand it. Do things, activities that bring you pleasure, I think is the main message.
And then the last question that we'll be able to get through as the group here is from Enrique Lopez. He asks, are there studies or indicators on what factors affect more than others on the progression of dementia?
Dr. Reynolds, did you want to start with that one, or?
This is a really good question. I think, of course, some of the lifelong activity engagement is really important for maintaining functioning. And certainly we can start things later on in life and, maintaining them, is helpful on a number of levels, but our lifelong engagement, both cognitively, physically, and socially are all very helpful. And we certainly know that some of the risk factors would be, or protective factors would be, educational attainment. And so if you're really engaged on certain topics, then it's nice to keep those things up. But again, it's a lifelong endeavor.
And I always say, exactly, it's engagement, not being passive, being active in things. And that's why if you are a hearing individual, protecting your hearing, but also just staying socially engaged. And lastly, brain health relies on heart health. Because as I said, there's so much vascular content. It just uses our energy. It uses our blood. There needs to be waste taken away. So heart health, brain health, and social engagement, and use it or lose it, your brain.
Ooh, that's a fantastic way for us to end. Yes, it's a all-around body health. I would like to thank Drs. Carson and Reynolds once again for a fantastic presentation this evening. And thank you to everyone who tuned in tonight. It was wonderful to have your questions.
Thank you very much for your engagement and your presence. Thank you.
Yep,
Thank
we
you very
look forward
much.
to seeing you on May 25th, for those of you who want to stick around for the last installment of our Exploring the Aging Brain series.